The atomic age was ushered in on July 16, 1945, by the detonation of the first nuclear weapon at the Trinity Test Site in New Mexico. This marked the successful completion of the Manhattan Project, a top-secret government project led by physicist Richard Oppenheimer. The primary goal of the Manhattan Project was to develop a new type of bomb based on a self-sustaining nuclear chain reaction involving uranium. We're talking nuclear fission, splitting the atom and releasing huge amounts of energy in the process. But this wasn't just about making bombs. At the same time, scientists also wanted to harness the power of the atom as an endless source of energy without those harmful emissions associated with burning fossil fuels. Well, radioactive waste can be a problem, but we'll circle back to that in another video. Living organisms get their energy by breaking and forming chemical bonds, not by nuclear fission. We build large, complicated macromolecules from smaller organic compounds, which themselves are made up of atoms. The word atom comes from the ancient Greek atomos, meaning uncuttable. Now, the idea of this word is generally true. Matter's granular, and we typically define it as having mass, taking up space, and consisting of atoms. But since the early 1900s, scientists discovered that atoms aren't actually the smallest unit of matter. Atoms are made up of protons, neutrons, and electrons. Neutrons and positively charged protons form the center or nucleus of the atom, with negatively charged electrons orbiting around it. Electrons are an elementary particle. They're a single unit of stuff, existence. But protons and neutrons can be broken down into quarks and gluons. Gluons are weird, though. They don't have any mass, kind of like photons or light. Gluons are just the particle glue that holds quarks together, and as a byproduct, the nucleus of the atom. But anyway, that's enough about particle physics. When it comes to the chemistry of life, the first thing we need to talk about is carbon. Carbon is a non-metallic element with an atomic number of 6. This atomic number represents the number of protons found in the nucleus of the atom, but the number of neutrons can differ from one atom to another. The standard atomic weight of an element is basically just the number of protons and neutrons in an average atom. For carbon, most atoms have 6 protons and 6 neutrons, giving them an atomic mass of 12 atomic mass units, or Daltons, AMU. But occasionally, a few carbon atoms will have 7 or 8 neutrons, so the standard atomic weight of the element's actually a little bit higher than 12. When atoms of an element have different numbers of neutrons like this, they're called isotopes: carbon-12, carbon-13, carbon-14. And this term was coined by Margaret Todd, a Scottish doctor, in the year 1912. But this classic illustration of an atom is actually wrong. Electrons racing around in distinct orbits like planets in a solar system, this is the Bohr model of the atom, but electrons don't actually circle around like this. Atomic orbitals take on a diversity of shapes and energy levels, and the exact location of an electron could be anywhere within these clouds. When atoms, or molecules, gain or lose an electron, they pick up an electrical charge. If an ion has more electrons than protons, it's negatively charged and called an anion. If an ion has fewer electrons than protons, then it's positively charged and called a cation. Ions with opposite charges can form ionic bonds, like table salt, NaCl. Each atomic orbital only holds a single pair of electrons. If an atom has unpaired electrons, it can form covalent bonds with other atoms, creating larger molecules. In a covalent bond, these unpaired electrons pair up, and the two atoms share them, all happy and equal. Normally, carbon has 2 unpaired electrons, which would mean it could only form 2 covalent bonds. But carbon's special. Its electron orbitals can hybridize, which rearranges the electrons inside them and results in 4 unpaired electrons. This means carbon can form twice as many bonds as before, and that's the basis for organic chemistry. Here on Earth, the conditions are just right for carbon-based life. But carbon isn't the only special element that can form 4 covalent bonds. Just Look at silicon, a metalloid with the atomic number 14. Maybe it's possible that on some other planet, one with incredibly high temperatures and a much different chemical environment, you might find silicon-based life. But on Earth, life is carbon-based, and it's dependent on water. So water must be a pretty special molecule too, huh? Let's take a look at its structure. H2O, oxygen, hydrogen, and a second hydrogen. It doesn't seem all that complicated or special. There are no unpaired electrons, no net electrical charge, but it turns out that water's polar. And when we say a molecule's polar, then one side has a slight electrical charge to it. Oxygen has a much greater electronegativity than hydrogen, which means that the electrons shared in a covalent bond between oxygen and hydrogen aren't shared equally. If they were shared equally, then that bond, or the whole molecule, would be considered nonpolar. In a water molecule, oxygen is selfish. It likes to keep the electrons closer to itself. That's what makes the molecule polar. We end up with a partial negative charge on the oxygen side of the molecule where the electrons like to hang out, and then a partial positive charge on the side with the two hydrogens, where we're not as likely to find those shared electrons. Because water is polar, this means it can interact with other charged compounds. Like dissolves like. It's a phrase you've probably heard before. This means that water acts as a solvent. Water molecules happily interact with ions or other polar molecules, while nonpolar molecules like fats aggregate together to avoid water. You've also probably seen how water forms a meniscus in a glass container. This climbing effect is due to adhesion, water molecules clinging to a surface. But as an example, let's take a quick look at how salt dissolves in water. Regular table salt is a crystal lattice of sodium cations, the smaller blue ones, and chloride anions, those larger green ones. In water, the ionic bonds are broken, and the ions dissolve in solution. These individual water molecules are oriented around each dissolved ion with their slightly positive hydrogens pointing towards the negative chloride ions, and the partially negative oxygens pointing towards the positive sodium ions. Since water's polar, it should be able to interact with itself too, right? In neighboring water molecules, weak bonds form between the partially positive hydrogen and that partially negative oxygen. These are called hydrogen bonds, and it produces a sort of sticky effect called cohesion. Even though hydrogen bonds are relatively weak and short-lived, about 5 to 10% as strong as covalent bonds, they are one of the underlying reasons for some of the other amazing properties of water. So let's talk about those special properties. With all these hydrogen bonds, it turns out that it takes a lot of energy to raise the temperature of water. This means that water has a high specific heat capacity. It holds a lot of thermal energy. Similarly, water has a high heat of vaporization. You have to put in a ton of energy to evaporate water, which is why sweating is such a great way to cool down when it's hot outside. Another interesting property is that ice is less dense than liquid water, and that has to do with how frozen water molecules form a lattice structure, and in that crystal, hydrogen bonding holds those molecules in a fixed orientation, keeping a fair amount of space between each one. In liquid water, the molecules are oriented in random directions, so they can get a bit closer to each other. This means that ice is less dense, so it floats. And it turns out to be pretty important from an ecological perspective too. Because ice floats, lakes freeze from the top down. So instead of the ice pushing all the fish and frogs and other critters up and out of the water each winter, they stay relatively warm and happy in deeper water. Lastly, water can form ions, protons, and OH-. These protons actually don't float around on their own. We end up with something like this. But anyways, one water molecule acts as an acid, donating that proton to another water molecule, and that second one is a base since it accepts that proton. This only happens occasionally and with a very small number of water molecules, but it has an important implication for biological systems because water can interact with buffers to keep pH levels within a constant range, homeostasis. Anyway, it's time to start building some biological molecules, the molecular basis for life. Most biomolecules are large polymers composed of many smaller subunits called monomers. Mono meaning one. For example, a polypeptide or protein is a long chain of amino acids. Amino acids are the building blocks, and they're linked together or polymerized through dehydration synthesis, which is the removal of water. But it isn't free, it costs energy to do this. So if we want to link together a bunch of monomers into a long chain, we put some energy in and we end up with a large, wet polymer. Now, if we start with a polymer and we want to break it down, we can do this by adding water. This molecule here was starch, and it's a long chain of glucose, sugar molecules. Now, this doesn't require any energy input since we're breaking high-energy covalent bonds and reforming lower-energy bonds. Essentially, we're releasing the energy that was stored in that chain of monomers. When we talk about cellular respiration and glycolysis, you'll see how different enzymes specialize in breaking down different materials and how breaking down monomers even further produces all of the energy that a cell needs. These polymers are called macromolecules, which pretty intuitively means big molecule. There are four main types of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. All types of macromolecules contain large amounts of carbon, hydrogen, and oxygen. Proteins also have nitrogen, and nucleic acids also have nitrogen and phosphorus. At least, those are the major elements that are present. Some types of lipids also have phosphorus. Some proteins contain sulfur. There's always exceptions. For the rest of this video, we'll be talking about carbohydrates, lipids, proteins, and nucleic acids in more detail. Okay, starting with carbohydrates. Carbohydrates are made up of simple sugars, or monosaccharides, that are joined by glycosidic bonds. Simple sugars follow the basic formula CH2O, and all of that is multiplied by N. For example, this here is glucose. Glucose has the formula C6H12O6. Ribose, right here, and also hanging behind me on my shelf, has the formula C5H10O5. If we just want to transport or store a few sugars, we can link them together, forming a disaccharide. Di comes from a Greek word that means twice or two. For example, sucrose is a disaccharide made from glucose and fructose. But if we take a bunch of monosaccharides and link them together with glycosidic bonds, we get a polysaccharide. Poly means many. This is useful for energy storage. Animals store glucose as glycogen and plants store it as starch. These polysaccharides can also be used in structural support. The cell walls of plants are made of cellulose and the cell walls of fungi are made of chitin. Okay, lipids. Lipids are kind of a diverse group of nonpolar macromolecules that include fatty acids, triglycerides, sterols like cholesterol, and phospholipids. Fatty acids are long chains of carbon and hydrogen with the carboxylic acid group at one end. If we put 3 fatty acids together, along with the glycerol, we get a triglyceride. Triglycerides are the most common type of fat found in vertebrate animals and plants. These fats can store twice as much energy as carbohydrates. But there's a ton of different fatty acids, which means there's a ton of different triglycerides too. But we can classify the different fatty acids into a few major groups. In a saturated fatty acid, like you'd find in animal fat, all of the carbon molecules are connected by single covalent bonds. In an unsaturated fatty acid, like you'd see in plant fats or oils, 2 or more of the carbons are connected by a double bond. When we look at this double bond, the hydrogen molecules can be on the opposite side of each other. This is the trans orientation, or they can be on the same side, the cis orientation. In the cis formation, we get a sharp bend here in the chain, which means the entire molecule is going to take up more space. Unlike saturated fatty acids, which can pack together pretty tightly, making them solid at room temperature, unsaturated fatty acids tend to be liquid at room temperature because of these kinks in the chain. One last type of lipid, for now at least, are phospholipids, like this phosphatidylcholine molecule I have here. Phospholipids are made up of two fatty acids, a glycerol, and a phosphate group. These fatty acid tails are nonpolar, so they're hydrophobic. They'll aggregate with other nonpolar molecules when in water. But this head here is polar. It's hydrophilic. It's completely happy being in an aqueous environment. Phospholipids can form lipid bilayers with the nonpolar tails pointing inwards towards each other and the polar heads pointing outwards towards that aqueous environment. This way, every part of the molecule is happy and it forms the basis of a cell membrane. Moving on to proteins. The basic building blocks of proteins are amino acids, and I have each one here marked with a label. They're joined by peptide bonds, and a short chain is called a peptide, while a long chain is called a polypeptide or protein. The peptide I have here is oxytocin. It's a hormone that's produced by the brain, and you might know it better as the love hormone. Let's remove one of the amino acids by hydrolysis and take a closer look at its molecular anatomy. This is tyrosine. It's a partially hydrophobic amino acid with an aromatic ring. Not only is tyrosine an important part of just about every protein in your body, but it's a precursor to catecholamines like dopamine and epinephrine, adrenaline, not to mention melanin, which is a pigment commonly found in the skin. All amino acids have an amino group at one end, and that's this NH2 with this blue nitrogen, and a carboxylic acid group at the other. All amino acids also have a unique R group or side chain that gives it its individual chemical properties. Tyrosine's side group is this aromatic ring. It has a hydroxyl group at one end though, so it does give it a slight hydrophilic quality, but remember, oxygen's pretty selfish when it comes to sharing its electrons. Anyway, different amino acids have side chains with different properties: shape, charge, polarity, acidity, and so on. Take cysteine, for example. Its side chain contains sulfur, this yellow one, and it's actually really special because it'll form bonds with other cysteines, and even if they're really far away on the polypeptide chain. These are called disulfide bridges, and actually we saw one earlier in our oxytocin molecule, and that's what kind of gave it that looped shape. The actual order of the amino acids is the primary structure of a polypeptide. But because of all those different unique properties on the side chains, we start to see some interactions between neighboring amino acids that form secondary patterns, like alpha helices or beta pleated sheets. But these bonds and interactions can happen a lot further down that chain too. In fact, disulfide bridges were a great example of that. This folds the protein into a complex three-dimensional shape or tertiary structure. In a few cases, proteins are actually made up of different subunits that come together, and we'd call that a quaternary structure. The actual shape of a protein, its tertiary structure or quaternary structure if it's made up of several subunits, that's what determines its function. Many proteins are enzymes, which bring together certain molecules to speed up or catalyze a reaction. But other proteins are involved in defense, transport, support, movement, storage, regulation, and some proteins even help other proteins fold the right way. They're called chaperone proteins. As incredible as proteins are, they're the reason living things are actually alive, they're not invincible. Low pH levels or high temperatures, for example, can cause them to denature. Denaturation causes a protein to unfold or fold incorrectly, which causes it to become non-functional. Okay, the last category of macromolecules are nucleic acids, which include DNA and RNA. Nucleic acids are polymers of nucleotides joined by phosphodiester bonds. Each nucleotide is composed of a nitrogenous base or nucleobase, a 5-carbon sugar, and a phosphate group. In RNA, the sugar is ribose, which is why we call it ribonucleic acid, or RNA for short. In DNA, the sugar is deoxyribose. DNA stands for deoxyribonucleic acid. There are 4 different nitrogenous bases in DNA. We have adenine, thymine, guanine, and cytosine. In RNA, thymine is replaced with uracil. Now, DNA is a very stable molecule. It structures the very familiar double helix with hydrogen bonds between complementary base pairs. There are 2 hydrogen bonds between adenine and thymine, and there are 3 hydrogen bonds between guanine and cytosine. The actual sequence of nucleotides, though, is what makes up the genetic code. RNA, though, it's not as stable as DNA. It's just a single strand, which is why if it gets too long, it bends and contorts to the point that it can start reacting with itself. Sometimes that's good. Some RNA needs to take on a specific structure to do its job, but it's not great if you're trying to store genetic information in the long term. The main job of RNA is to read DNA and direct protein synthesis, but that's the topic for another video. And actually, that wraps up this video. So thanks for watching, and I'll see you next time. These polysaccharides can also be useful for structural support. Not this one. The cell walls of plants are made of cellulose, and the cell walls of fungi are made of chitin. Wow, that was the most ridiculous timing.